the synthesis procedure, which probably once we have light tomorrow, we are going to start from there, just to lay the foundation. At least we should know what we are expecting, what we want to be doing before we venture into it. So this slide is just to show us little things about what synthesis is all about, okay? So I'll just give you an idea of what and what do we expect, how do we go about it. Then when you now enter into the synthesis room, you will know and what it entails, what you want to do, okay? So these are the procedure for this presentation. So let's go to the next slide. So it will cover this scope, nanoparticle, which we have laid the foundation in the first presentation. Then the synthesis protocols. We want to look at the protocols that guide synthesis. Then we look at the characterization techniques. What are the various characterization that is necessary or needed after synthesis? What do you do with your data? How do you characterize it? So we are going to be dwelling more on that one. Maybe tomorrow, Wednesday, Thursday, we're going to look at all those characterization techniques. So I discovered that many students, they only go to the lab, they will synthesize. They don't know how to bring the data into figure. They find it difficult. I have many students, even master students. They find it so difficult to bring the data into figure. All those figures, I was showing you the other time. They are all data. You must learn how to produce figure from what? From the data. So if you don't have the requisite knowledge, that's why I was saying that we should download that origin. We're going to be using the origin and image so that you will master it. And once you know it, you know it. There's no two way about it. It's very simple to operate. All you need to do is to put you through, and once you get the requisite direction, then you can go ahead and whatever data you have, you can plot your data, you can manipulate your data anyhow. You can manipulate your data with origin. You can manipulate your data with origin. So it's very important. We will not rush it. We will take it step by step so that we can master it. We can do some calculations like crystallized size. We heard about it. If you have a sample, how do you get the crystallized size? How do you get the particle size? And we are telling you what is the size of this sample, what is the crystallized size of this one. So we are going to calculate them one after what the other. So we get some data, then we are going to use, then we conclude. Next slide. So, as I've said the other time, nanoparticle can be from the organic level and can be from the what? Inorganic level. We have nanoparticle that if you synthesize it, you will know that you can obtain it from the organic level. And what we are discussing here, as a result of this workshop, we are talking about renewable. So anything that is renewable is something that is accessible. It's not exhausted anyhow. Then the organic aspect of it has to come in. And in the course of the synthesis, we are going to work more on that organic one, so that we can see how we can use what we have in our vicinity to actually, obtain good results. One of us in our research group, Dr. Omaya, she won a grant, and what she did was she used green synthesis approach, to fabricate supercapacitor battery. That's why she won flare grant. She won that fellowship, and she only used organic approach of synthesis for supercapacitor for battery, and that gave her that millions. She was able to buy equipment from that grant, and she's doing fine with the grant. So we can actually explore more on this organic aspect of what? Of nanoparticles. Organic aspect where we have green synthesis, biological synthesis, and they are not too costly because they are affordable. You can access them easily, and they are renewable. You can't exhaust them. Don't need to ship in all those expensive chemicals before you can synthesize. In fact, most of my work has been on biological approach of synthesis. So we can easily do it and we can easily publish it. Very easy and direct. We can use it for diverse application, for supercapacitor, for battery, for biomedical, for energy. You can use green method. Very direct and simple. And if you learn it, once you master it, you master it. There's a guy now that is still calling me, I think from which university now. He says all he needs is the synthesis procedure. He attended one of our last conference, and since that time he has been calling me that I should just give him the procedures. But you are privileged today because of this workshop. The procedure has been has been prepared. It's not easy. It's something that you should pay for. You don't just give procedure to somebody like that. Are you getting my point? But the center has paid for this, so I align my procedures. Once you get the procedure, all you need to do, master one, replicate the other. Are you getting my point now? Once you master one, you can use one to produce many. One thing about publication is that once you break the gist for one paper, just try your best, break it. Once you can publish one paper in a good journal, subsequently, I don't think you have problem. You just discover that you work on that one, then you begin to what? To produce more papers. So please let's just follow it step by step so that we can learn. Because after this workshop, we want people that will have potential in synthesis, people who have potential in what? In characterization. Then you can easily go to your lab, synthesize, and after characterization, analyze your data by yourself and beginning to write paper from it. Very important. So you don't need to wait for anybody. All you need to do, look for a viable lab where you can synthesize. Another challenge we are having is this. Many people find it difficult to formulate projects. Design is very important. Research design. You know, this research design has kept many students in school for years. Especially when you see a student that is not working in line with the supervisor. When there is no synergy between you and your supervisor, you discover that that student can stay for 2-3 years. What is he looking for? He's looking for what? Design. How to design that research. Research design is the most important. Once you get it done from the design, the next thing to do is to go to the lab. You know what to do. But once you don't have what to do, there is no way you will struggle. You discover that you are just buying chemical or pump chemical. You go to the lab, you put something together, no result. You go back and design. So research design is very important. So before you go to the lab, you must what? Sit down, read articles, read current journals, try and put something together. Design the what? The work. Once you design it, it will be much more easier for you to go to the lab and work. Very important. So research design, very key to whatever you want to do. Then once you have it done, in the lab you can, 1 week, 2 weeks, you can be done with your experiment in the lab. But you can spend 2 years in research design. Don't ask those PhD students. When you don't get it right from your design, it will be very difficult. So let's go ahead. So both organic and inorganic. Organic, you use many things, surfactants that you can use for your organic. So we have done this in the morning. That is nanoparticle, whose size is measured in nanometer. The particle has different shapes. And the shape is very important for your application. If you know what you want to apply it for, then you know what you want to get. Either spherical shape, whether rectangular or any shape. Rectangular, tubular shape, we have flower-like nanoparticle. In biomedical, mostly, they prefer a spherical nanoparticle because of the transport application of it, so that it can easily move within the system. So they prefer spherical nanoparticle. So that's why most nanoparticle for biomedical application, they prefer it to be in spherical shape. So it's easier to work with spherical materials than the. So we look at then can exist also in fused form. It can as well be aggregated or agglomerated form. So we have different shapes of nanoparticles. So it depends on what you want to get. And agglomeration is somehow, to avoid it, there are some techniques you have to follow during synthesis to avoid agglomeration. That is the essence of, I brought ultrasonicator. It also helps. It will help you to purify your sample. It will also help to avoid agglomeration. When you ultrasonicate your sample very well, it will purify your sample, and it will also reduce the agglomeration level, so that everything will not just fuse together. You will see particles, they are distinct. You see some work, you will see the particles are very distinct. They will separate themselves very well. You see the nanoparticle. That shows that work is very pure. Maybe it has been well ultrasonicated, and you see them very distinct. So these are the work of some of this equipment. Let's go ahead. So, okay, we are talking about nanoscale. Nanoparticles are particles that have one dimension in nano range. Now, if you look at this range now, the nanometers, the molecules. So you can see where your nanoparticle falls in. This is colloid, virus. No, we use microscope to be able to see virus, right? So you can see where they fall in. See where bacteria falls in. So this range, that's where nanoparticles falls in. So you can almost have the same range with virus, micro. So molecules at this level, micro at that level. So you can see the range. Well, very important at that range. That's what we are studying. They are very, very minute. That's why they can easily transport themselves within the cell. Just like your organisms. If you have all these organisms in our system, in our blood, they can move easily to anywhere. The same size. Nanoparticle is as smaller as all those viruses in our system. Are you getting it now? See, virus can move. Even in the brain, we have a brain, all those systems, they can move anywhere in your body. So nanoparticle can as well travel within your system, just the way virus can travel within your system. Okay? So let's go to the next. Now, this nanoparticle, after synthesis, after characterization, then you have something in mind. What am I going to do? Characterization, the separation techniques. If you look at this, these are separation techniques that are very important. Which one? I said it the other time, to avoid agglomeration, there must be what we call separation techniques. See what I mentioned the other time is one of them, ultrasonication. You have to ultrasonicate it very well to avoid agglomeration. That's the separation techniques. So many people use chromatography, use ultracentrifugation. You can also centrifugation. You can also use it as a means of separation techniques. Then the characterization. Now, after you have obtained your nanoparticle, what do you do with them? You have to characterize, and these are the major things you can use: your AFM, atomic force microscopy, STM, SEM, scanning electron, TM, the transition electron, XRD, DLS, and UV, FTIR. It depends on your application. Then you now be talking about what characterization should I do. If you are working on energy, you know that you are going to do your CV. If you are working maybe on another thing, your dielectric properties or conductivity. I think Madam was talking about the stress part of the material. You want to look at the tensile stress, the strain, the micro strain of it. It depends on which angle do you want to tailor the material? Then you go ahead and look for equipment or machine that can characterize it for you. There's no material that you cannot characterize. It depends on which application. And so far, that material can be used for that application. That is the essence of research design. You have to look at it. Is this material good for this application? Once it's good for that application, then you sit down and design. Let me design it. And, when you are designing, you want to avoid plagiarism. You want to look at, okay, this work, has it been done? That is why you have to subject it to some time. You have to try this work. Somebody has done this in this way. How will I replicate this work in a way that will be a bit different from what this person has done? That is research. If you use A and B, you want to use A, B, and C, and try and compare your result with what he has done as A and B. If you are in the same line, you have contributed to knowledge. That's what is going on in research. So basically, you have to look at it. Then quantitation techniques. These are the equipment you can use to quantify your sample. You have to do a lot of the quantitative measure. Then the impact of it, a lot, which you can actually, it depends on you, either in energy, you can make your impact in energy, in agriculture, in medicine, you can apply it. And these are the types that we are using. We have carbon-based nanostructure. Some people are working on carbon-based, where you can use graphene, use other things, carbon-based. Fluorines, carbon nanotube. It depends on what you want to use it for. Then we have metal oxide nanoparticle. That one is very common. I think we use that one often. We use metallic oxide, we use zinc oxide, we use iron oxide, titanium, magnesium oxide, iron, silver. These are things very common which we have used. We have worked on silver, we have worked on zinc, we have worked on magnesium, we have worked on magnesium, zinc, nickel. We have worked on virtually all these metallic oxide. And the quantum dots as well, metallic nanoparticle, which is like gold, like cobalt, like zinc, like copper. We have also worked on it. My interest is mostly on ferrite materials. Ferrite materials are very good for biomedical applications because they has an element of iron in them. So we tailor it for what we call super paramagnetics. We want to look at the magnetic property of that material. So that is why we use it mostly for biomedical application because of that ferrite nature. So it has some magnetic properties, which if you want to use it for drug delivery, it will be very effective. If you want to use it for hyperthermia, it will be very effective. If you want to use it for photodegradation, it will be very effective. If you want to use it for energy, very effective. You can use it for dielectric properties and purposes. You can use it for it. Very effective. So I've been working on ferrite mostly. I work on zinc ferrite. I work on cobalt ferrite. I work on magnesium ferrite, nickel ferrite. It's a ferrite family that I often work on, both the inverse and inverse type of it. So basically, this is what nanoparticle is all about. Next slide. Now let's look at the novel properties of nanoparticles. What makes nanoparticle unique, or a novel one that people are talking about now? Number one is small size. Look at the size at even smaller than the so-called virus in terms of size. Very small. So it can easily move into any system. Nanoparticles, the size give it one of the highest advantage because of the size. So anything they are doing now, it has to pass through the nano range. Everything, even our memory card, They are in the nano range. All manner of things. I show you that nano watch the other time. You can imagine what the battery will look like, what the memory will look like. That nano watch has memory at a very high rate. It has memory. You can imagine the battery, lithium-ion battery that can have higher energy, that can store energy. It's not a battery that, because if you use all the ordinary battery, it won't last now. Because the application inside that watch, the applications are just too numerous. So you can imagine it's on 24 hours. It's working with internet. Just the way you insert your SIM in your phone, that watch is working 24 hours. So you can imagine the kind of battery that watch will be using. It won't use this ordinary battery. I think Dr. Nwaya is working on that. He has successfully done something on battery. He was able to pelletize it and have lithium-ion battery at a nano range, very small size, that can be used for different applications. So that's one of because the size matters. The second one is what the high surface area of nanoparticle. High surface area is very important, and that's what makes it very unique. Then is ease to suspend in liquid. Easily suspend in liquid. Can easily diffuse in liquid. Then if it diffuse in liquid, it can easily move. Not that it will find it difficult to diffuse in liquid. Easily diffuse. So that's one thing that harms it, a better application. Then deep access to cells and the organelles. Deep access. Easily, it can easily migrate or move within the cells. It can move within the cell just the way your blood is flowing all through your cells. And the particle can diffuse into the bloodstream and move all through your cells easily. So it harms it, another good application. The improved physical, chemical, and biological properties. The physical properties, the chemical properties, the biological properties, all of them. We have to test it. It tests the biological component of it, the toxicity aspect of it, the bioassay. We have to do it. Then to know if this thing is good for this particular application. So all those things are necessary. So properties of nanoparticles are different from their bulk counterparts. You cannot compare with the bulk one. Bulk counterpart is different from the nano counterpart. So let's go to the next slide. Okay. So if you move down to the synthesis techniques, this is where we need to learn some things. The synthesis techniques. There are two major approaches to it. The top-down approach and the what? The bottom-up approach. Top-down approach comes from a bulky material down to, I think Prof said it in the morning, the Prof that came in the morning, you remember. So people are moving from very large, complex system and they want to achieve something. No, nanoparticle means you are coming from the smallest one to achieve a purpose. Okay? You can imagine, you combine just small nanoparticles to build something, a robot. Somebody was telling me in the morning that some country that they build a drone, form of a fly. This is our normal fly. This is a butterfly or housefly. And you are just seeing housefly flying up and down. You don't even know. You will be thinking it's ordinary fly. They are not ordinary fly. They are drone. Drones everywhere. And those drones has ability to shoot, to kill. On man. These are as a result of what? The nano-size range of those materials. You can imagine they use drone. They have been using like US, whatever, using all those unmanned drones to kill people. If they are targeting you, they will just use unmanned drone. They will just send it. You will be thinking it's bird flying. You will be thinking it's bird. That bird that is flying may be a drone flying towards you. You won't even take cognizance of it, and you'll be thinking that you are enjoying the bird flying. It's targeting you. Once you are alone, you release the target. You are gone. So it's as a result of what? Nanotechnology. So we are coming from, if you look at this one, it's becoming bulky. People are no longer working on this. You have the physical method. Even the physical method, the equipment are very, very expensive. People are not even patronizing them because it's only those big countries who can afford those equipment that can still be working on something like this, bulky material coming down to nanoparticle. But we want to come from those from the atomic level, gradually build something that is useful. Okay? If you come there, so that's what we call bottom up synthesis approach. Then under that bottom up synthesis approach, we have the chemical approach, we have the biological approach. We can easily work on some chemical approach or biological approach because this synthesis techniques is basically in 3 dimensions: the physical approach, the biological approach, and the chemical approach. Okay? So the chemical and the biological approach mostly are from bottom up approach, while the physical method of synthesis is mostly from top down approach. Okay? So we are going to see some different method under this approach. Go to the next slide. Okay, see them. So top down approach. We have the chemical etching, laser ablation, mining process, sputtering, thermal decomposition, lithography. See now all this approach. How many of them do we have here? I doubt if we have any one of these. You can only get all this approach in all those big universities where they, like my supervisor, my PhD supervisor, he went to Göttingen in Germany, University of Göttingen in Germany. His own area, this is what he used, sputtering, in analyzing his own sample. He used sputtering method because everything is there. They have it. This sputtering, laser ablation, it's not easily, but it's not a thing that we can do. Now, if you look at the bottom-up approach, you will see some method that we can easily, do, like the chemical vapor deposition, the CVD. We can do that easily. In our lab, we do that. The CVD, then soil gel approach. We can easily do that one. It's a bottom-up approach. Then we have a laser pyrolysis, spray method. You can do that one easily. Molecular condensation, the aerosol process. Then it's divided into 2, which is the green synthesis. Green synthesis involve the use of extracts from plants, from fruits, from different things. We can use it. So anything, when you check the phytochemicals and they are very good, you can use it for synthesis. You have to be very careful about what you are using. So all these are vegetables that are edible. Some of them are very good. You can use them for synthesis. We have used bitter leaf, this moringa, even all these many things are using. Scent leaf. There's one they call Grongonema latifolium. That one is also very good. I've used it. Tazi, very good. I've used it. We have used bitter leaf. We are using moringa. We also use many expert spinach. So there was one that interested me when I started. The person used watermelon ribs. And when I read through that article, that was my first interest in that green approach. Use watermelon. I said, watermelon? Okay. What did he do? He just the back, that green aspect of it. He peel it and dry it and turn it to powder. And use it for green synthesis. And I was looking at the results. Very wonderful. I said, wow. So this thing can be useful. I saw one from Lautech. They use a cocoa pod. Cocoa pod? Cocoa seed. They got the cocoa pod, they blended it and turned it to cocoa powder and cocoa something. And they use it to synthesize good nanoparticle for biomedical approach. It was very nice. They gave them very good results. They published it in a very good journal. Since that time, I have some interest in all those things. So it can also help you when you pick one or two things from this aspect. So you don't need to carry too much load upon yourself that I want to do laser blazer. It must be laser blazer. When your colleagues are doing small things and they are getting 100 paper, 200 paper, when promotion time comes, they file promotion. They are moving, pump. You are still racking your head on what to do on sputtering, and you have not breakthrough for one paper. You'll be there. Are you getting my point? Look for something you can do that can project you up fast. Something you can do. So lay your hands on experimental protocol that's very fast and you can do easily, and the resources are what are available. Look at our environment. That's what I used to tell myself. I will look at the environment. I look at what we can do within reasonable time. Not that if I give you a topic and I know that this topic you cannot finish it. There is no way you can finish this topic. Why am I wasting my time? There's no resources, no place to characterize this. Even the one we are doing here, we will still send it to Canada, send to South Africa, send to this place for characterization. We don't have the way we talk. All the characterization are not complete. In the entire country now, I don't know if we have any place we have TEM. I have not seen TEM. Yes, TM. Do you have it? Footmina. They have TEM. Footmina. TEM. Or SEM. They have TEM. That would be good if they have TEM. You mean? Okay, it's dead. It's not working again. So and TEM is very expensive. TEM. So these are factors, and all this equipment are supposed to be in not even in institution, in each department. In fact, chemistry department is supposed to have their own SRD, SEM, TEM. Go to physics department in the same institution. They have their own equipment. Not under normal circumstance. In a very good country. Only that we don't manage our... We are good. Yes, I'm saying we are good. The entire country, no single functional term. As big giants of Africa. There are no single term. And we say we are trying. We try, but our try is not enough. When you enter a particular country, you enter this university, just enter their lab. Go to bioengineering. All these equipment are there in bioengineering. Go to chemistry. All of them. They don't borrow equipment from bioengineering. No. They have it. The government knows. They finance it. They give them grants. What do you want? I need the social equipment. I was in NUST. NUST is one of the best universities in Pakistan. It's a new university. Very compact. They invited me to give a talk. I went there to give a talk. So I entered their lab. They did introduction. I was wondering. Only one man attracted VSM. VSM vibrating sample magnetometer for characterizing magnetic properties. Just one of their staff got a grant from government and he bought VSM, put it in their lab there. VSM, each time I want to characterize, I used to see, I have to contact people in China, in Pakistan, come and help me. Oh, there's one man in Iran that want to help me. I send my sample through DHL. Do you know that they turned my sample back to me? My money is still hanging now, almost 50,000. The DHL money. They said, Iran, they are fighting. DHL cannot deliver my message to them. After they have collected my money, I'm still writing now to refund my money. You can imagine how terrible is this. And these are samples that we are not even sure when the result comes back, it will be good. We are just working on that probability. I get to my point. And yet we are trying our best to produce something. But you are in a lab whereby the VSM is standing, SEM is standing, term is standing, SRD is standing. I walk into my sentences room. Sit inside. I will take it, the sample, UV, wrong. If it's correct, if it's not correct, I don't like the result. I will go back, re-strategize my procedure, change the techniques, the procedure if necessary. I will go back and test from UV. I will use my SRD to test. Not that now we will try biochemical. You will sit aside. You will now post it to South Africa for SRD. If your result is good, you are fortunate. If it's not good, that procedure is a waste. You start all over again. Are you getting my point? So you can see where we found ourselves. It's a serious matter. It's a serious matter. But I know God is helping us. We are still trying our best in all this. So, are we here? Okay, go back. Let me see if I'm done with this. Okay, I'm done with this one. So I think I will be rounding off in this stage so that tomorrow morning, go to the next slide, then I will round up this. So we are going to start with something like this tomorrow morning. So we are starting with soil gel. Soil gel approach is a very simple approach. If you look at this, very simple approach. But let's just stop there so that tomorrow we'll start from here.